Now that we've seen the anatomy of the immune system and the main characters involved, we can look at their roles in host defenses. The second line of defense includes inflammation, phagocytosis, interferon, and the complement system. Now, these are relatively nonspecific in their effects, but they do communicate with the third line of defense. Those interactions we'll look at in our next chapter. First, let's begin with the inflammatory response. An inflammatory response is a reaction to any traumatic event in the tissues. It can be burns, scratches, bites, or lesions, or even allergies. And all of us manifest inflammation in the same way. When the inflammation is close to the surface, we can identify a classic series of signs and symptoms. These symptoms are rubor or redness, which occurs due to increased circulation and vasodilation at the site of injury. Calor or warmth. Naturally, if we have increased circulation and vasodilation, there will be increased heat. So this is given off by the increased flow. We'll also see tumor or swelling. The swelling occurs because of increased fluid escaping into the tissues from the local vessels. And then dolor or pain occurs because of the stimulation of free nerve endings, which is sometimes due to the stretching of the tissue. Now, a fifth symptom has been added in order to make a more complete story, and that is loss of function. Often we'll lose function due to the swelling. These symptoms may be really unpleasant, but they do serve an important role as a warning system to set in motion a response that prevents further injury or infection. And as you're probably aware, in some cases, inflammation can affect an entire system, like the entire lung or the entire circulatory system. There are several chief functions of inflammation. First, mobilization and attraction of immune components to the site of injury. Then we set in motion the mechanisms to repair tissue damage and localize and clear away harmful substances, and then destroy the microbes and block further invasion. The inflammatory response is a really powerful defensive reaction, but when we have chronic inflammation, you probably have seen it can cause tissue injury, such as in the incidence of cardiovascular disease, serious inflammation of the vessels because of the deposits in them. So cardiovascular disease is actually a result of chronic inflammation. Now let's look more at the stages of the inflammatory response. The earliest changes occur in the vasculature in the vicinity of the damaged tissues. Many of these changes are controlled by nervous stimulation and involve chemical mediators and cytokines released by blood cells and tissue cells and platelets in the area of injury. Here we can see the mast cells releasing chemical mediators. Some of these chemical mediators are vasoactive and affect the endothelial cells and smooth muscle cells of the blood vessels. Others are chemotactic factors or chemokines. These affect white blood cells and may draw them out to the site of injury. Inflammatory mediators cause fever, stimulate the lymphocytes. They also prevent the spread of viruses and cause allergic symptoms. First, the arterioles constrict rather quickly, but then they rapidly release to a state of vasodilation. This vasodilation allows increased blood flow to the area of injury. In the next stages, we see edema, or leakage of vascular fluid into the tissues. The fluid that escapes through the gaps in the walls of these capillaries are called exudates, and the accumulation of these exudates causes edema. The exudates are illustrated in yellow here in the figure on the right. The exudates contain plasma proteins as well as blood cells, fibrinogen, which is a clotting factor, and cellular debris. They may be clear, which we call serous, or they may contain red blood cells and pus. Now, pus is simply white blood cells and debris that's generated by phagocytosis, so it makes sense. So you might ask, how do these blood cells make their way out of the vessels and into the surrounding tissues? The process is called diapedesis, and it involves white blood cells adhering to the walls of the small vessels and then squeezing their way out between the endothelial cells. How do they adhere to the walls? Well, the endothelial cells of the walls have some adhesive receptors, and then the white blood cells stick to those adhesive receptors and literally squeeze themselves out through small perforations in the endothelial cells because they can change shape really easily. And then you might ask, what causes the white blood cells to move out of the blood vessels? That's where chemotaxis comes into play. There are specific chemical stimuli that are released at the site of injury that call the white blood cells into place. So literally, we'll see because of this chemotaxis that white blood cells swarm from other compartments to the site of inflammation. That way, they're ready to take care of any further invasion if it should ensue. So what are the benefits of edema and chemotaxis? First of all, both the formation of edemaceous exudate and the infiltration of neutrophils are physiologically beneficial activities. The exudates will dilute toxic substances. Also, they contain fibrinogen, which will form a fibrin clot. This clot can trap microbes and prevent further spreading of the invasion. Phagocytosis occurs immediately as the neutrophils exit the blood vessels. And sometimes we'll see formation of pus because certain bacteria are pyogenic, that is, pus forming, because they attract extra neutrophils to the site of injury. Sometimes mild inflammation can be resolved by edema and phagocytosis. However, if there's a long-lived inflammation, it'll attract a collection of monocytes, lymphocytes, and macrophages to the reaction site. The macrophages are the trash truck guys, so they clear up the pus and cellular debris as well as any dead neutrophils and damaged tissue. Concurrently, we'll see B lymphocytes coming to the site of injury, and they'll produce antibodies. Those antibodies can then attack any invading pathogens. And then we'll see T lymphocytes killing intruders directly. And then the final stage in the process of inflammation is that tissue is repaired or replaced by scar tissue or other connective tissue. So now that we've covered inflammation, let's cover phagocytosis. Phagocytosis is the cornerstone of inflammation and specific immunity because this is where all of the byproducts get gobbled up. It's generally accepted that all cells have some capacity to engulf materials, but not all of them are professional phagocytes. These guys do it for a living. The general activities include surveying the tissues and compartments and discovering microbes, particulate matter, or injured and dead cells. They ingest and eliminate these materials. And when appropriate, they'll extract immunogenic information or antigens from the foreign matter in order to boost our third line of defense or our specific defenses. There are three main types of phagocytic cells. There are neutrophils and monocytes and macrophages. Now, monocytes, as you are already aware, become macrophages and they move into tissues. While they're in the bloodstream, though, they're considered to be monocytes. In the bloodstream, they'll mature and then get themselves into some tissues where they become resident or roam around. Most of them that roam around are roaming around in the reticuloendothelial tissues. Some of the resident macrophages will become resident in places like the liver or the lungs. So as we examine the stages of phagocytosis, remember that there's more than just the physical process of engulfment. And this process can happen as either a single incident or as an orchestrated large-level event for inflammation. We've already discussed the idea of chemotaxis. The phagocytes are attracted by a gradient of stimulant or chemicals to the site of inflammation. Some microbes can be recognized directly because of signal molecules on their surface. These signal molecules are called PAMPs, or pathogen-associated molecular patterns. PAMPs are not present in mammalian cells, so they act as red flags to the cells of our innate immune system, that is, our non-specific immune system. PAMPs are things like peptidoglycan or lipopolysaccharide or double-stranded RNA, things you wouldn't see in mammalian cells. On the host side of the equation, our immune cells have PRRs, or pattern recognition receptors. These pattern recognition receptors do exactly that. They recognize the PAMPs and launch an immune response. Now, here's an example of a PAMP. They're toll-like receptors, not because they're anything to do with payment or anything, but the original receptors were toll receptors, and these ones are toll-like receptors. They're surface receptors on the macrophage, and they bind to particular PAMPs, or molecular patterns, on foreign molecules. The signal is sent into the nucleus, and then the nucleus will work to produce cytokines, interleukins, and other inflammatory mediators to launch an inflammatory response against this foreign molecule. Pretty neat system. Now, as for the stages of phagocytosis themselves, we have the engulfment or ingestion phase. This phase may involve PAMPs and that kind of recognition, but either way, we'll finally have engulfment of the pathogens into a vacuole. This vacuole is now called a phagosome. So once we've engulfed the whole thing, formation of a phagosome occurs. And then there are lysosomes. You'll recall that all the lysosomes in the cell contain degrading chemicals. They're sort of like the recyclers in the community of the cell. These lysosomes will then fuse with the phagosome to form a phagolysosome. In this phagolysosome, there are enzymes like lysozyme, DNase, RNase, proteases, and peroxidases that act to break down the invading bacterial cells. Also, we'll see that destruction occurs through a respiratory burst. This respiratory burst released reactive oxygen products like superoxide radicals and hydrogen peroxides. This burst of oxidative agents degrades the particles further so that they're no longer a hazard to the cell. After the phagocyte is done breaking down all the chemicals, the process of exocytosis occurs, and all of these particles are released from the cell in the process of waste elimination. Let's take a look at a quick animation of the process of phagocytosis to help us better understand the process.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
Before we move on, let's have a quick concept check. Which step of phagocytosis do microbial capsules interfere with most? Now this one's asking you to take it a little further out. Think about what chemotaxis is, engulfment, lysosomal fusion, and oxidative killing. Chemotaxis is where chemical signals are released at the site of inflammation. Does that have anything to do with microbial capsules? No. So we can cross that one off the list. Engulfment. That's where the microbe is taken in by the phagocyte. So it's a good possibility. Lysosomal fusion. That's where the phagocyte has already taken in the microbe and a lysosome will fuse with the vesicle. And then oxidative killing is even further degradation. So I'm going to cross both of those off because engulfment is the most likely here, as the microbial capsule could probably interfere with the binding of the microbe to the phagocyte. This is why encapsulated microbes are such an issue for our immune system.